So
thank you, and thank you for the invitation to speak. I'll just going to sort of reflect a little bit on my experiences in designing clinical trials and conducting clinical trials during the ongoing COVID pandemic. Now, the particular challenge that we were facing, or that we are facing, with any new disease is that it brings many uncertainties. Yet at the same time, especially in an infectious disease, what we need to do, what we must do, is get the trial started as quickly as possible to identify useful treatments. On the one hand, of course, because we want to help the patients, but that's true for any trial. But in the context of a pandemic, of an outbreak, it's particularly useful to be quick, because a useful intervention is going to also be part of the outbreak response. In other words, if we can identify quickly a treatment that is going to help patients and reduces infectiousness, we can at the same time reduce the size of the outbreak. Now, for COVID-19 in particular, a challenge is that the presentation of the disease is quite heterogeneous. A lot of cases are asymptomatic. If you have symptoms, they can improve without any major issues within days, but it can last weeks or even months, and obviously also lead to death. Now, looking back a little bit to the 2014 Ebola experience, I think there are some very interesting lessons to be learned already. So what you see in my graph here are the number of new Ebola cases over time. On the x-axis, you got time. On the y-axis, you get the number of cases. And the two different shaded lines, different colored lines, are basically just two different reporting mechanisms of the number of cases on a given day. So you can see the outbreak started around May 25th, and then it peaked around the 9th of May. Now, what's interesting is that the arrows indicate when different clinical trials have started in Ebola at this time. And I'm going to argue that some of them, with hindsight, of course, never had a chance to provide an answer. If you look at the last two, ZMapp and TKM, at that time, the number of new cases was so low that it was going to be very, very unlikely that they'll ever recruit enough information to make any decisions about the utility of that, of a particular treatment. But even for whole convalescent blood, it just about reached a peak. And for me, that is a clear indication how important it is to get a trial started, and to get a trial started quickly. Now, I got into the lucky position to be able to influence and get involved very, very early on in the COVID outbreak. And in particular, the first trial that I was able to contribute to was the Lotus trial. And that trial started on 18th January 2020. Now, if we look back, if we think back, you may remember that the first sort of confirmed case, or initial first confirmed case of COVID, at that time not even known under that name, was on the 31st of December 2019. And so 19 days later, this trial started. So clearly, with that, with the trial, we've achieved one of the things that I already claimed and argued that is important, i.e. be fast, get ready quickly. Of course, that's easy said and more difficult to be done, especially if you want to apply the sort of typical rigor, the typical standards to a trial that you would sort of usually want to use. So there were a couple of simplifications, pragmatic choices that we had to make in that trial. So for example, rather than the trial being a placebo-controlled trial, it was an open-label study. The treatment that was used was lopinavir-ritonavir, which is an HIV treatment. Effectively, it was a treatment that was readily available and had some preclinical evidence that it might be useful in COVID as well. But clearly, it wasn't the only choice. And in fact, there were several other choices that were discussed. The other rather pragmatic choice that was made at the onset of the trial was the primary endpoint to use. So the endpoint of the trial was the rate of clinical improvement on this 7-point scale that you can see on my slide here, up to day 28 or hospital discharge, whichever happened first. And clearly, because at that point, there was very, very limited information about COVID and the presentation of COVID. This was heavily informed, the choice of this endpoint was heavily informed by other infectious diseases and influenza in particular. And finally, you can see at the bottom of my slide, I'm going to argue that the trial started without any formal design. And by that, I mean, there was no sample size calculation done. The priority to get the trial started quickly was get the machinery in place to collect the data in a randomized fashion, and then worry about how many observations you will need. So with that already, I think it's clear that the trial started and we expected modifications or refinements to the study from the onset. And so the trial was subsequently adopted within the first week or two of running. So the first and probably most notable difference was a change in the primary endpoint. It was from a rate of change to a time in clinical improvement endpoint. We did also conduct a formal sample size calculation and discussed various sort of more complicated, more complex trial designs, such as group sequential designs, that is, trial design that allows us to stop investigation early, either because the treatments look very promising or because the treatment don't look very promising. And we also discussed, in the light of the fact that we were considering a handful of potential treatments in the first place, we also discussed the possibility of including several active treatments in that trial. We didn't go forward with these more advanced designs. Instead, reality almost overtook the trial. So after we've recruited roughly half of the patients that we intended to use, there was evidence coming out that an alternative treatment, remdesivir, might be more suitable and better, more useful for COVID. And so the decision was made to stop the trial and instead focus on remdesivir. So the second trial, which started in early March of 2020, in fact, the second pair of trials, was investigating remdesivir as a treatment for COVID. And the two different trials focused on mild and moderate cases versus severe cases. In both instances, a group sequential design was used, and you can see at the bottom of my slide here, a schematic of what a group sequential design looks like. On the x-axis, you have the number of patients that you have observed the primary outcome from. On the y-axis, you have your test statistic comparing your treatment against control. So basically what you do is, after you've observed N1 patients, you compute your comparative test statistic and check, am I in the red region on my graph? In which case I would abandon, I'd give up with the conclusion that sorry, it doesn't look like that treatment is going to provide any benefit. If I'm going to be in the green region, I'm going to pop out the champagne and celebrate because I've identified a treatment that significantly improves whatever my primary endpoint is over my control group. And if your test statistics happens to fall in that white space in between, your conclusion is, sorry, need more data to make a conclusion, and hence you would recruit further patients and repeat the exercise after having data for N2 patients until you reach a conclusion. So for both of these trials, the one interim analysis was planned, and that interim analysis was planned at the halfway point of the trial. The primary endpoint was time to a 2-point improvement, very similar to the lopinavir-ritonavir trial, except it was using a 6-point scale rather than a 7-point scale. And that's simply because we've learned from the first study that we couldn't reliably distinguish the bottom two categories, category 1 and 2, on my previous endpoint. Additionally, because of concerns that the treatment might not be as effective if given too late, we restricted recruitment to be within 10 days of symptom onset. So that trial recruited up until the point where pretty much the initial outbreak in Wuhan waned and faded, and the severe trial was stopped and reported after, again, roughly half of the patients that were originally planned, just about the time when the interim analysis was planned, the trial reported. For the mild and moderate trial, the trial, technically speaking, is still ongoing. Numbers are very, very low still, because at the time of the big outbreak in Wuhan, basically the resources, all resources were focused on the severe cases, and so it was very, very difficult to find the staff to actually also recruit on the milder end of disease. So at that point, we started to realize and think about, well, it doesn't look like, unfortunately, that COVID is going to remain contained within Wuhan or the Wuhan region, but instead it started to branch out and expand into other countries. And so we started to think very hard about what we consider what the perfect trial during a pandemic could look like. And some of the things that we came up with included: it uses a meaningful and efficient endpoint, it discards poor treatments quickly, it can cope with multiple treatments, and it minimizes the burden on frontline staff. And obviously there's a long wish list, and I'm sure everybody here on the call can probably add 1 or 2 other things that they would like to see in a perfect trial during the pandemic. But for us, these were the sort of 4 main aspects, in addition to the realization that whatever the trial design looks like, it will have to be adaptive. It will need to be able to be modified on the basis of what we learn as the outbreak continues. So with that, we then set out to design the recovery study, which is a multi-arm platform trial that initially investigated 4 active treatments against standard of care. The primary endpoint was mortality at 28 days. And in order to minimize the burden on frontline staff, minimal data collection was used, and most of the data, or a lot of the data that is being collected within that study, is obtained through data linkage. And I'm going to say that so far, recovery has been a great story. In the first 90 days, it recruited about 11,500 patients. And to date, it's just over 40,000 patients recruited. As of today, there are 181 sites in 4 different countries. The trial is open to patients of all ages, from neonates to over 100-year-olds. And probably most impressive, it only took 9 days from the funding decision to recruiting the first patient into the study. So here is how the initial phase of recovery looked like, how recovery unfolded initially. So the trial was opened on the 19th of March. We get standard of care against 4 active treatments: hydroxychloroquine, dexamethasone, lopinavir, and azithromycin. And the choice of treatments was informed by a priority list that was established by an expert group linked to the WHO. And in addition, it was a requirement that the treatments needed to be scalable. So it needed to be enough treatment available so that it could actually influence the outbreak as it was ongoing if it proved to be effective, and it needed to be available in large enough quantities to be trialed in a large number of patients. About a month after the trial started, an additional treatment was added. Tocilizumab was added for patients on the particular severe end of treatment. Another month later, convalescent plasma was added to the trial. And a week after that, the first result of the trial was made public. And that was hydroxychloroquine not providing any mortality benefit in hospitalized patients. Another 10 days later or so, the first successful treatment was identified in dexamethasone in patients on the severe end of disease. And another couple of days later, the conclusion was reached that lopinavir was not going to be effective. As you probably have heard and know, the trial has gone on and has so far provided answers to 9 questions, showed that 3 treatments reduce mortality, at least in some patients, in some subgroups of patients, and at the same time provided conclusive evidence that 6 treatments did not provide any mortality benefits. It was recently named as the trial of the year by the Society for Clinical Trials, and it's since been used to model or influence several other UK-based, well, not just UK-based trials, but also UK-based trials such as Protect-CH or Heal COVID. And as of the last couple of weeks, there have been discussions of using similar models outside of COVID and use a similar setup to recovery in other diseases. So that's all a great story so far, but I think we always should reflect on whether all that looks good actually has been as good as it can be. And I think there are still a number of interesting questions that where we can improve upon the design, upon the recovery trial. And chiefly, the room for improvement comes from 3 different areas. The first is around treatment uncertainty. So that is the question about how many treatments am I going to include, which treatments am I going to include, and when do I include a new treatment in the study. Now imagine that we would have lived in this fortunate world where after the first couple hundred cases, the outbreak in the UK would have waned, we would have been in a situation where we wouldn't have answered any questions. If we had included several more treatments, the chances of answering any of these would have been even lower. And so it is not as trivial of a question of, oh, I'm just going to include additional treatments in the study, because the number of treatments also means that the number of patients that I need to recruit, and therefore the number of patients that I can recruit, needs to be large enough. And during an early stages of an outbreak, we just don't know which way the pandemic is going, or the outbreak is going to go. The other question that was discussed quite heavily within the recovery group, for example, was how should we include treatments into the study? So, for example, several treatments have been included in a factorial manner such that patients might receive multiple active treatments. And it is not clear in which circumstances, in which cases, this is the right thing to do always, and in which cases it would be better and clearer if one would only randomize patients to a single treatment rather than giving them the option to be included in multiple treatments. The second question is around the population size, and I've already touched on that, and that's the question of how big are the number of patients going to be that are going to be affected by the outbreak, by the disease. Now, for recovery, that meant that the original trial did not include any formal stopping rules. It did not include any formal sample size calculations. And instead, the decision about, or the task of making recommendations about stopping treatments was given to the Data Safety Monitoring Board. The final point, which I'm going to spend a bit more time on, is the uncertainty about the endpoint. Now, I've started off talking about the Lotus trial, which used initially a rate of improvement endpoint that was later switched to time to improvement. The recovery trial used mortality as the endpoint. But especially early on in the disease, it is very much unclear what the right kind of endpoint should be. And so, thinking a bit about the choice of primary endpoint, we found the statement in a paper from a few years ago by Whitehead and Horby quite interesting, in that it said that in the context of an infectious disease, the outcome of recovered versus not recovered is almost always of interest. So that's easy, right? I mean, it's either recovered or not recovered, dead or not dead. So these kinds of endpoints are meaningful, but also at the same time, they have the challenge to go with them that they tend to require very large sample sizes. And as I've indicated very early in my talk, we want to be quick about making decisions about treatments, and hence having to recruit thousands and thousands of patients actually might be contra the intention of having answers quickly. An alternative is to have an ordinal scale of some sort, which effectively takes the recovered versus no recovered and puts a bit more granularity into that endpoint. Or we could think about biomarkers as some quantitative measure that we think might be indicative of clinical benefit. Now, in the context of COVID, it wasn't obvious at the onset what a useful biomarker would be. And it was also felt that for the more confirmatory type studies that I've been talking about, you wanted a bit more hard endpoint that measures a clinical benefit, and hence I'm going to focus thinking about the ordinal scale and the recovered, not recovered mortality type endpoint subsequently. So the first question with these ordinal endpoints is, how are we going to analyze these endpoints? So the obvious choice is to say, I'm going to fix when I'm going to look at the distribution of these categories at a fixed time point, for example, at day 14. And that's been used over and over in different infectious diseases and has been shown to be a very useful endpoint in this context. The challenge with a new disease is we just don't know what the right fixed time point is. Is 14 days after treatment, is that too late? Is that too soon? We just don't know. And so there is a question about, what if we get the timing wrong? Secondly, there's the statistical assumption of proportional odds that's often used in this context. And finally, it might be difficult even for statisticians to appropriately interpret what an odds ratio or a log odds ratio means. An alternative could be time to improvement or time to recovery type endpoint. The drawback there is we effectively dichotomizing our outcome. So it's only recovered or not recovered, or has it improved sufficiently, yes or no? And so that we know is going to lose information. At the same time, contrary to just using a binary endpoint, it takes the time course into account, and hence we're going to gain information through that. One nice feature about the time to improvement, time to recovery type endpoints is that you can use incomplete information, and that's particularly helpful if you want to undertake interim analyses. Basically, what that means is patients who haven't already been in the study for, say, a month still contribute to the analyses. The final point is, how do you deal with patients that die? Using a time to improvement endpoint is not an unsurmountable problem, but it requires a bit of thought how to do that. Now, obviously, one could also simply sum the categories across the study days, for example, through the total sum of scores, which, again, it might be difficult to interpret this outcome. It will be a very powerful outcome because it uses a lot of information, but interpretation is difficult. If I'm going to have a too long observation period, then I'm going to dilute any treatment effects. Imagine I'm going to look at the sum of the scores across 100 days, for example. You would expect that most patients after the initial couple of weeks would have returned to their sort of normal state, and hence you would be adding up a lot of the same numbers, and hence you would reduce the differences between the groups. So the duration matters in this case. And you're going to have to fight with missing data in this context. So in order to understand a bit better what the implications are of these different ways to analyze and use different endpoints, we conducted a small simulation study where what you can see here is on the x-axis the days since randomization. The sort of the faint lines correspond to 100 sort of generated outcomes of individual patients, and on the y-axis you have their ordinal score going from 1 to 7, just like we had on the initial lopinavir-ritonavir trial. The thick lines correspond to the effect in the placebo or in the control group and in the active group, respectively averaged across all patients in the study. And so here in my little illustration here, there's a bit of a difference between the active and the placebo group, and that difference starts to emerge right at the onset of receiving treatment. So in our simulation study, what we've done is we've investigated a number of different scenarios. So the one that I've just shown you here in my table is called the reference scenario. Then we have a scenario where basically the pattern is the same, but it starts a week after treatment initiation. Then we have a pattern where patients recover faster. We have a pattern where there's a higher mortality rate. And the final scenario is a case where there is no difference other than the mortality rate. The different columns in my table here correspond to different ways to analyze our potential outcomes. So the first one is our ordinal scale using a proportional odds model, either at day 1, day 7, day 14, or day 28. The mean score, that's just the average score over 28 days. And then there are different ways of looking at time to outcomes. So time to a 2-point improvement, time to recovery, that is discharge from hospital in this case, and time to death. And finally, the last column corresponds to mortality at 28 days as the primary outcome. Now, the first thing to note is that whether you use a proportional odds or a mean score or a time to event approach, generally speaking, the power, the chance of showing a difference when it is there for all these methods is going to be higher than if you use just mortality in the last column. And similarly, by just using time to death, I'm also going to lose some power. And so including these different categories actually is helpful in order to increase our chances to find useful treatments. Now, the other thing to notice is I've already talked about these two factors that influence the time to improvement, time to recovery type endpoints. On the one hand, I'm dichotomizing and hence I'm losing information. On the other hand, I'm increasing information because I'm considering the time course. And it appears that in combination, these two together mean that I'll end up roughly at the same level as if I were to use a proportional odds or a mean score approach. So then looking a bit more closely into the results, I think the most interesting finding is, as we feared, is if there is a lagged treatment effect. And so here, just remember, no changes for the first 7 days, and then the two groups start to separate. If I would have used a proportional odds model with and looked at it at 7 days, I would have gotten it horribly wrong. My power would have basically been minimal, just at 5%, while other approaches give me up to sort of 85% power. The other thing to say is that pretty much throughout these evaluations, you can find that the time to endpoints are doing very well in all the scenarios. No, they are not always best. No, but they're also never much worse than the best of the outcomes. And hence, my take-home message from that little evaluation was that the time to recovery or time to improvement outcomes are a robust choice that give you high power, more power than using mortality. Now, clearly, there are some other differences. If I'm talking about time to recovery, the interpretation of that outcome, for example, will be very different than if I'm talking about a mortality benefit. I'm not going to take that away, but if I'm just looking at both of them being, thinking about both of them being potentially useful outcomes, I think the choice is pretty clear that the time to recovery endpoint is a better choice, a more robust choice, a more powerful choice. Okay, so that's just gives you a flavor of what we've done in the recovery trial. Now, recovery is in particular initially focused on repurposed treatments, but at the same time, we also recognized that it's going to be important to also think about novel treatments that have been designed specifically or optimized at least specifically for COVID. And so we also designed Agile, which is an early phase platform trial with the objective of undertaking dose finding together with some initial proof of activity in a sort of seamless phase 1-2 trial design. For this trial, we've used a Bayesian framework in order to maximize our ability to find signals. I'm not going to spend much time talking about the details of the design, but here is a schematic of what the trial looks like. We enroll an initial cohort of patients. We record the outcomes for safety initially, and if there is safety concern, then the study is terminated on the basis of safety. If it's deemed to be safe, then we estimate what the next dose ought to be for the next group of patients. And so we might either continue with the same dose level or we might increase the dose level. At the same time, while that decision is made, the patients from the first cohort will be observed for their efficacy activity endpoint, which is either time to improvement or time to viral clearance. And a decision is made once the data for that treatment are available, for that dose are available, whether or not it's worthwhile continuing. So there's an interim analysis for benefit as well as futility. If the decision is need more data, additional patients are recruited, and so on and so on. So then just to sort of come to an end, a couple of thoughts about what to do if an epidemic wanes. Now, I'm going back to the Ebola example because actually that work has started already as a consequence of the 2014 Ebola outbreak. And as I've already indicated before, here, my take-home message from that little graph is that basically all the trials started too late to have a viable chance of providing useful answers. Now for COVID, as of today, there are over 1,200 trials recruiting worldwide and 75 trials in the UK. And so just the sheer number of trials mean that there's a high risk of underpowered studies, in particular if the outbreak wanes. And so a consequence of these underpowered studies is that a potentially useful treatment might be discarded because, well, in that underpowered study, it didn't show sufficient promise. And so in order to avoid, we have thought about the idea of ensuring that for diseases that occur in outbreaks, we avoid underpowered study through a collaboration approach. So effectively, the thought is actually quite straightforward. The trials use a core protocol. And if you get to the point that you cannot recruit any further patients or its recruitment is so slow that it doesn't make sense to continue recruiting to your study. You use adaptive methods at the end of the outbreak to make a decision, first of all, whether or not you have already enough information to reach a conclusion. If the answer to that question is no, you keep the results blinded. If there is no definitive result, and at the next outbreak, you would restart that protocol and hence augment the sort of data from the new outbreak with the information from the old outbreak in order to get convincing evidence for any treatment under investigation. So just to wrap up, I think COVID has shown that well-conducted randomized clinical trials are useful. They are the gold standard for evidence gathering outside of a pandemic, but also I think COVID-19 has shown that they also are during the pandemic. I believe that for any trial in a new disease, the trial design needs to be adaptive, simply because there are so many things that we learn as the trial goes on that it would be foolish not to include that information if it is helpful. Mortality, as shown in recovery, is definitely a very desirable primary endpoint, but it may be difficult in practice to use because recovery, with hindsight, as the trial was ongoing, decided that it needed around 2,000 patients in each arm in order to reach conclusions about treatment. So that requires recruitment of about 4,000 patients, which, okay, with COVID, unfortunately, we got into the position where this was possible. But for many other diseases that occur in outbreaks, this is possibly not going to be feasible. And if that's the case, then time to improvement, time to recovery endpoints, I believe, are very useful alternatives to consider. I think we're seeing now that the focus is shifting away from repurposed treatments. And in order to enable that, one also needs to think about how one can make sure that these confirmatory studies, such as recovery, do have a pipeline of treatments coming in. And we've tried through the Agile platform to provide that, that pipeline. And finally, everything I've talked about would not have been possible without very close collaboration. And COVID has shown that many of the hindrances and hesitations that we often see in traditional trials can be overcome very quickly, and collaborations that otherwise might have been difficult have been shown to work very well. And so I think a lot of what I've talked about would not have been possible without these collaborations. Thank you.
All right. Thank you, Professor Jaki, for a wonderful talk. It's very impressive. So it looks like there's a few questions coming now through the chat. One from Mark Pilling. Do you have any comment on the additional risks of trial speed and trial complexity? For example, I guess the AstraZeneca's misdosing in Argentina, one dose showing higher efficacy than two.
Yes, so there's obviously always a risk of trying to get answers quicker, right? And so I think the important aspect is that of my learning, I guess, is that a lot of the delays that we do see in sort of traditional clinical trials have nothing to do with the scientific rigor. They have to do with contracting. They have to do with requirements on monitoring, review processes and things like that. And these processes can easily be reduced without compromising the study integrity entirely. At the same time, of course, yes, there are certain things that where there is an increased risk. I mean, for recovery, for example, we have very sort of minimal safety monitoring in comparison to a traditional trial. Now, here the argument is made on the basis of that these are well-known treatments, and hence the side effect profile is well understood. But obviously that is not appropriate all the time and everywhere. Talking about other studies, I mean, there've been plenty of other studies where positive results have been shared, even though that the studies were heavily underpowered, were of poor quality, and so on and so forth. So yes, I think the standard argument of well conducted, I think I've said explicitly here in my discussion slide, it's important that these trials remain well conducted, and so shortcuts should not be on the quality of the study, but on the sort of administrative hurdles, in my opinion.
Great. We have another question in the chat from Amy Mason. With respect to leaving underpowered trials blinded and restarting recruitment again in the future, are there any potential negatives for the trial participants in doing this?
I don't think there are any obvious negatives for the trial participants. I mean, it is a challenging concept because it implies that the results of the study might lie dormant for a long time, right? There is a risk of information leakage, of course, because somebody will still know the results at the interim analyses, and that information may well come out. So that's not trivial to handle. There is a risk, I guess, of sort of negatively influencing the maybe not negatively influencing, but maybe more undervaluing the valuable work. Imagine that you are responsible for the data collection in the first trial, and then 5 years later, your trial gets completed through a second outbreak. You've essentially delayed showing what you're capable of by 5 years, which obviously isn't trivial, and especially in an academic world. So there are definitely challenges to overcome, but I think it is still better than not than reporting trials that are underpowered.
Great. Another question from Stephen K. Can you comment on any challenges in real-time randomization across multiple sites and potential considerations for clustering by specialty of hospital, for example?
So that aspect, to be honest, I was only peripherally involved. So there are probably other people that can comment on that better. But one of the reasons, for example, why we have gone with simple randomization in recovery was to avoid any additional complexity in the randomization process. Now, I think having we have the tools available and the online randomization systems nowadays that you could do something more complex, even across different sites and so on and so forth. But in the interest of simplicity, in the interest of avoiding potential problems, we've gone for the simple option here, and that is simply to avoid potential hiccups.
Great. And another question from Mark Pilling again. do you have any comment on the place of observational data analyses as trials during epidemics, i.e. Hernan's work, alongside the gold standard randomized clinical trials?
I think observational studies are potentially very useful. I think what the risk isn't so much in the utility of these studies, it's how they are interpreted subsequently. Ultimately, an analysis from an observational study will always contain increased risks of bias, and it will never be at the same level of evidence as a randomized study. And so as long as that realization is clear, as long as that is obvious, as long as the results are interpreted in that way, I think there's a great role of observational studies. In particular, I mean, we've seen in the vaccine studies, for example, that effectively the post-marketing surveillance, what importance that has played, for example, and that would not have worked without sort of useful and good observational data analysis methods. So I think there is a big role. I don't believe that they will ever replace a randomized clinical trial, but as the question was phrased, it's supplement and add to, and for that I think they're extremely useful.
All right, thank you. I think we're pretty close to the half hour, so I'm happy to leave it there. If there are no more questions or if there are any more comments you want to make, Professor Jaki?
No further comments from my end.
All right, great. Well, thank you everyone for coming. And I'll be in touch for the future seminars, which will resume after the summer. Thanks, everyone.
Thank you.